Hi everyone. Before we start the session, we are inviting you to look at the question we put on the screen. You can continue to talk, but this is just to give you a sense of what we had in mind when we built this session. And we're going to start, I guess, in a few minutes, or shall we start now?
It's up to you.
Okay, I think it's okay. So welcome everyone to this session on impact of scientific advances in plant health. My name is Odile CARISSE. I'm a plant disease epidemiologist from Agriculture and Agri-Food Canada. And Claire Baker is from, well, she's going to introduce herself, but she's the co-chair of this session. We don't have much time for the introduction. Just want to say that when the scientific committee discussed about the different session, we thought that it would be interesting to take some time and zoom out a little bit and look at the impact of what we are doing as research scientists. And Claire would say on the real world, so the impact on plant health. So let's start with the first speaker. Claire Baker will be talking about one aspect of the Toothpick Project that she's involved in. So Claire.
Hi, thank you for coming to our session today. I am not a scientist. I majored in art history, so I'm the only one here, I'm pretty sure, that probably majored in art history and doesn't have an advanced science degree. But my partner and my colleague is also my dad, David Sands. So some of you who've been in this field for quite a long time have met him probably over the last 50 years as he's been coming to these meetings. He's a plant pathologist, professor emeritus at Montana State University. And it takes all types to get to impact. So my 30 years in NGO development fit very well with his biotech innovation. And so we sort of met along the path, and I'll talk you through that today. It's called the Toothpick Project. It's a biological herbicide. And that little purple flower you see on the left there is, I think it's on the left, yeah, left there too. That's called striga or witchweed, Kayongo. It's a weed across Africa. It's on 40 million farms. I'll be talking about that in the framework of impact. We look at impact with a variety of different things. It's not something that you can look at siloed, or we choose not to. So I'm going to break it down today in discovery, humanity, economy and policy as sort of two joined things, and environment. So discovery, in my head, I'm thinking of it as the science, the research, and also prior to getting to that, perhaps it's an understanding of the problem that needs to be solved. And I'm thinking of that as part of discovery. So in the case of the Toothpick Project as a biological herbicide, this is my dad's history, 5 decades of work. I'm actually not going to go through it. It's tiny little print, but I'll certainly send you this slide if you're interested, and papers on each of these things. But it was his discovery along the way, looking at different pathogens that can kill weeds, looking at amino acids that can inhibit plant growth, and then eventually merging those into a single solution. And this is where we pick up in 2007, which is striga. This is a field, a maize field, obviously not very successful with maize. Striga witchweed is tackling it, and it's a parasitic invasive weed. It attacks the roots of maize, sorghum, millet. It's dryland rice, sorghum, sugarcane. So basically, it's depleting crop yield on 40 million farms. It'll deplete crop yield from 20 to 100%. If you're in Africa, you'll hear of it, and it has a lot of different names. There's some curses that go along with it, which is very interesting in the terms of impact. As we're developing a product, how do we develop science that also we can then communicate with the farmers as something to address a curse? It's considered the worst pest threat to food security in Africa. You can add up all the locusts and the fall armyworm and other pathogens and things, but striga tends to have the most consistent, constant crop devastation across the board. it's $9 billion in crop loss. That's off the tables of smallholder farmers, so it's a very interesting approach, whereas other crop loss that we're looking at is a large agricultural problem, and it's a lot easier to measure the value of it. And also, I don't have it on this slide, but 85% of maize farmers in Kenya are women. So this is a women's impact and also solution. I'm not a scientist. I've learned how to talk about this fairly well to a certain level. I'm sure you can all ask questions to stump me, but I can challenge some of them, I think, as an art history major. I'll run you through just the basic science of it, and we'll certainly can send you papers on any of this and have calls with you if you'd like to get more into the virulence enhancement process. But we are one of the first biological herbicides to be commercialized in the world. There have been maybe 24 or less other biological herbicides that have had various levels of commercial success, and some of them have dropped off of the commercial field. So this is a really exciting development because of the timing of where our commercial availability came, as well as some of the other conflicts that we've been seeing with chemical herbicides. So we're using Fusarium oxysporum forma speciale strigae, which is host-specific just to Striga. In fact, it's only specific to some Striga, not all. There are 40 different kinds of Striga, so or more. So Striga eromantica and Striga asiatica are the most common ones, and our strains will work on those. Wild type is not effective enough. It only will kill maybe 40% of the Striga in the field. So we are merging, this is my dad's innovation, to merge the natural pathogen with the ability for amino acids to inhibit plant growth. And he's using the Fusarium as a manufacturer of the amino acids. So he's basically selected the outlier spores that are excreting high levels of the specific amino acids that we want to use to kill the weed, but not the crops. And so we've selected those, and those are the strains that we're using, and then we had to figure out how to commercialize that. This is an important part, and I think it's something where my dad's quite unique actually in being a research scientist, but he always was thinking about who's the farmer that we're trying to solve the problem for. So from the very beginning, this research started in 2007. They were always working in the field with smallholder farmers. The product that they ended up developing, which is why we have this weird Toothpick Project name, is because we grow the fungal strains on toothpicks, then we can transport those to the village level very easily. We use these like Nalgene bottles. At the village level, we train village inoculum producers to cook a pot of rice, transfer it into a sterile bucket that we provide in their kit. They drop some of the toothpicks in, and then they seal it up and shake it twice a day for 3 days, and all of the rice becomes inoculated with the fungal strains. And then on that third day, they have about a week for it to get to the farmer, and the farmer just plants a pinch of it with each maize seed. And I'll show you some yield photos later, but it's really remarkable. We did a proof of concept trial with the Gates Foundation over 2 years, and that sent us on the path to commercialize this. We started the commercialization process, which is regulatory trials, in 2015, and then we finally got commercial registration with the Kenya Pest Control Product Board in 2021. So it was a long haul, and there were all sorts of errors along the way, but we finally did it. And then once we had that last year, we conducted a survey with our farmers. How are you liking the product? How is it working for you? Do you have any suggestions? And they said, kind of smells, it's kind of sticky. We don't like the short shelf life because it's the 7-day window. A few said it was too expensive. I was surprised. I thought we'd get more pushback on the cost. And quite a few of them had the interesting suggestion that it should be a dry product and it should be a seed coating. So we happen to have already had that sort of in the research bin. And so instead of coating toothpicks, we started testing coating wood dust. And this is with Dr. Peter Lutz. So many of you know him. He's our partner in this project as well. And we coat a wood dust, a sawdust, and then we can package that with some sugar. The farmer can then dump the spore powder with the sugar into some water, put the seed in there, their own choice of maize seed. It's resistant to Captan. It's not resistant to all of the fungal strains and fungicides that are sometimes coated on the seeds. So we have a list of what's appropriate seed to use. And then the farmer can just lay that out on a tarp in the shade, and once it dries in like an hour, they can plant. So this innovation we thought was going to take about 2 more years to get through regulation, and fortunately we got a surprising letter in June saying that it was approved. They couldn't see the difference between a toothpick and the wood dust. So that was very exciting, and now we've been hustling to get it commercialized for this growing season. And it's about 60% less expensive to the farmer. So that's part of our impact. So as we look at this, one of the sections that I was breaking this down into is environment. So environment includes flora and fauna and soils and water, but it also includes greenhouse gas emissions that if we're looking at replacing a product. So if we're replacing glyphosate, we can look up the studies for what are the greenhouse gas emissions of glyphosate. Also, we can replace tillage, which is used as a weed management system, and so that can help store some of the carbon. We also can look at biodiversity when we're looking at environment, and right now the farmers, a lot of them are monocropping, because you want to get your maize first and foremost, or your sorghum first and foremost, and then if you get that, then you can diversify. You might grow some cash crops, and also some crops that might have more nutritional diversity. So solving this problem also can impact biodiversity by giving farmers more choice and also nutritional diversity. As we're looking at the environment, we're looking at these other innovations and interventions and measuring what we're doing against what they're doing. And if we're replacing a product like imazapyr-resistant maize, we can look at what are the negative consequences of imazapyr-resistant maize, what's the impact of that versus what's the impact of ours. And that isn't just about like the greenhouse gas emissions or toxins, but also about the use for the farmer, and that's something that's important to us as a farmer-focused organization, because farmers have to plant IR maize with gloves, so that's a barrier for them, for their success. I'm not going to get too much into those. Economy, that's our next level of impact. This is one of the easier ones to measure for us, because we can track how much we sell, and then we can do some spot checks on how much improved yield they have. And it's very difficult for us to put a blanket statement on, like, everybody's going to get 50% yield increase. That's not the, whoa. I don't know what happened, but hold on. Anyway, that yield depends on how much striga they have. So they could have 100% loss, and then they'll get a 20-fold increase in yield, or they might just get a 15% increase in yield. So we can't. So we have to measure this. But here's some of the results from our. You can see there on the left that plot graph. That's the Gates Foundation over 2 seasons. That was the long range 56% increase in yield. You can see physically what it looks like with the maize. This is what the farmers see. They see their cobs. They don't care about a graph. They see their cobs growing. And the photo on the right there is Jeffries, and she's holding, like, physically in the field. She can see where she's treated and where she hasn't treated. Here's another demo plot of treated and untreated. And here's one from our partner World Food Program. This was last year versus this year. So when we're looking at economy, we're measuring those kinds of yield increases. We're also looking at the value that we're providing at the village level, and part of our manufacturing system is it's very important for us that we're having this driven by the farmers at the village level. So our economy, we're measuring how much the farmers are increasing. We're measuring how much the village inoculant producers are selling, and they make 54 shillings per unit. So they can make, I just got a text message a few minutes ago saying that somebody had just made $500 in sales because they had a good testimonial and they brought in their whole, I don't know, their whole church group or something. And so that's a huge amount for them because they're able to then send their kids to school. We also have in-house manufacturing where we can sell to agri-vets, and then we also can license to seed companies, which this is our first season with this new technology, so we haven't gotten to that point yet. But all of this will be measurable impact. And then humanity, this is of course, as a humanitarian, where our whole group is driven by this. This is the economic value, but then measuring what does that do at the human level. So what are the farmers choosing to do when they make an increase in yield? And there's something called, well, there's the Women Empowerment Agricultural Index. It's a measurement by the USAID. It's really complicated. In fact, I applied for a grant for like $100,000, and they said, no, you have to do women's empowerment measurement, which would have cost us about $30,000, I found out after I attended the session. It's like, oh, that's ridiculous to pay 30% on measuring impact. But, and now, since then, they've kind of streamlined that process. But this is, you know, we're looking at, are they making choices to send their kids to school? 100% of our farmers, pretty much when surveyed, I think it's 100% that they say they'll send their first income will go to send their kids to school. Medicine, phones, other farm inputs. So that's all part of women's empowerment. I don't see a timer. What am I doing? I don't know.
A few minutes.
Okay, great. So policy, and you see here I have the SDGs on the left. Hopefully you're all familiar with those. They're the Sustainable Development Goals. And this is sort of these were created as a universal way to measure impact. And under each of the Sustainable Development Goals, they also have indicators and ways to measure. And those are things that are supposed to be driving governments as they're setting policy. There's a little bit of a disconnect, which I'll get to in a second, but what we did when we first set up our company in 2016 was we had a stakeholders meeting. We hadn't even set up the company yet, and we invited the NGOs, the farmers, funders, the international agencies, research institutions, government, local organizations, seed companies, and it was a fantastic meeting. There were about 45 of us in Nairobi, and it was couple days long, and it was terrific, and we were like, this is our plan, we've got this. We forgot to invite the policy people, which turns out was a mistake, but there's this sweet spot, and I think we finally have nailed it, but it is an ongoing problem. You know, the Venn diagram, you've got policy, and those policy people are making decisions on what impact they want to see. And then you have the finance folks, and they're making decisions on what impact they want to see. And those don't necessarily connect to each other. And then you have the knowledge, which is the scientists, discovery, research, what do we know? That doesn't necessarily feed into the others either. So there's this big disconnect. We've been able to navigate ourselves into that sweet spot as a small, nimble organization. But I think that part of my responsibility is to talk about it all the time. So constantly talking about, you know, the finance, we cannot keep writing grants for $100,000 for a year if we want to actually make long change in agriculture. We need to also be talking about policy. You can't be making laws that you want things to change, like Sri Lanka, they banned herbicides, chemical herbicides, but they didn't have a solution. So then the knowledge folks were in a bind and the farmers boycotted. So all of this stuff has to be in sync. We've happened to be kind of in a lucky and also conscientious space where we've been thinking about how to link them all together. For us, this is sort of how this Venn diagram has looked for us. Finance, we need funding for research, of course, because we have scientists in 12 other countries that are ready now that we've successfully commercialized in Kenya. We need regulation because every country in Africa has separate regulation. We need a little bit more familiarity because people don't understand the host specificity, most importantly, as a key part of our innovation. And we need some scientific infrastructure. This entire market, herbicide market, is chemical right now. There have been other biocontrols in insect and fungicides, but not much in herbicides. So we just need to be doing more infrastructure as far as like getting approvals, that sort of thing. But we have great partners. We have heightened consumer awareness. We're very consumer driven. And there are no other alternatives. You can kind of skip this. This is sort of our network. And it's really, these players play a role in all of that policy, that knowledge, and that finance. So we've been weaving them together to develop impact. And we all do have shared impact goals, so that's an important part. And then just quickly, this is our vision for the future. So if you want to talk to us about what we're doing, our next steps will be working with we have scientists in 12 other countries, but Ethiopia, Uganda, and then Cameroon, Benin, Congo will be kind of our next areas investigation. We have some small grant funds for that. And then continue working on collaborative space, and then hopefully by 2025 start seeing our next regulations approved. Here's our science team. Here's our Kenya pilot team and our community advisors, including Cindy. And definitely reach out to us. That's it? Done? Yeah, reach out to us because we're very collaborative and constantly building our team.
So we have 1 or 2 minutes for questions in the room.
I see a couple here on the app. Yeah. Is the product commercially available outside Africa, and on which weeds? No. So part of it being host specific, it's only available on Striga. It's only available in Africa. We have actually just started a company in the United States called Kuvu, which means fungus in Swahili, and trying to fundraise for that, but we're apparently too early for all the investors. But that is going to be looking at other weeds. We've got some samples for thistle and bindweed kind of in the queue, and Orobanki. So we'll get there, but people don't get what we're doing yet. The next question is, isn't it risky to base protection on a single solution? Is there a risk of Striga developing resistance to fusarium? Absolutely. So first of all, no one asked the chemical companies this, and so we've seen herbicide resistance, which I forgot to mention in our discussion of environment. Herbicide resistance is one of the largest problems in the chemical herbicide world. So, but they're still on the market. They're not getting pulled off of the market because of herbicide resistance. They're getting pulled off the market because of toxins. So, yeah, of course, we are definitely aware of herbicide resistance. We've selected for 3 different amino acid excretions, which gives slightly more workaround. And if we can continue working with regulators, ideally, we'd love to see introducing new strains every 5 to 10 years to try and stay ahead of that kind of adaptation. So it's something on our mind. I think when we're talking about impacts, it's like you have to think about that forward. Negative consequences or unintentional consequences.
Thank you. I think we're going to move to the, thanks a lot.
I got you.
Want me to introduce it? Do you want to introduce that now?
Okay. All right, so thanks. Next up is Bernard Slippers from South Africa. Just flew in for this and over the weekend. Go ahead.
Thank you. Claire, thank you very much. I think your talk is a perfect platform for me, for my talk, where I'm not talking about a specific scientific invention of myself, but rather a reflection on our advances in science related to plant pathology over the past century. I'll talk about where that century comes from in a bit, and how we have moved forward in terms of managing diseases, and maybe what are some of those barriers and things that might be missing, and how that relates to one of the central tenets in plant pathology of our plant disease triangle, or as I refer to, the pyramid. Just recently, my co-author Eva Stuckenbrock and Sarah Gurr had a commentary in Nature where they reflect on the crisis that faces food production. And throughout this conference, there's been quite a bit of reflection on that. And they call on more involvement from government and industry, but also point out that it's no longer good enough to rely on simplistic approaches to breeding or to management of diseases or the use of fungicides. So there are elements that are missing despite all the scientific advances that we've made in terms of plant pathology. And I was reminded of this very vividly in 2020 when I was asked by the South African Journal of Science to write a commentary on the first plant pathology professor's address almost 100 years at that point. His name was Paul van der Bijl. So I read his address and was absolutely fascinated by the story and reflecting on just how far we've come in terms of plant pathology. At the time, they knew there were things that caused viral-like diseases, but they didn't have an idea about what those are. And if you think about where we are today in terms of understanding how individual base pairs within a gene really affects virulence, you'd understand how far we've come. But as I point out in here, it's sobering to reflect that despite all of those advances, we've declared an International Year of Plant Health because of the plant health crisis that we face. And that brought me to this reflection on, reading this paper on how Paul van der Bijl then reflects on these elements of the disease triangle. So there are many more words. I'm just grabbing some sentences out of his address, referring to the biological races, the variation in the pathogens and how that affects disease development, the variation that there is within the plant material and how that affects disease development, and the environmental factors that affect it. At the time, the disease triangle was not conceptualized as we know it today. That came in the 1960s. R.B. Stevens credited with first drawing this, and today we know this in all the classes that we teach, and in much of the work that was presented here this morning's keynotes reference to the disease triangle and these interactions between pathogen, host, and the environment. That conceptual framework is quite important because it influences how we're thinking about managing diseases, how we're thinking about projecting future impacts. A recent paper, this is a 2022 Trends in Plant Science paper by Yang and colleagues, reflecting on models of how plant diseases will respond to climate change, and pointing out that many of the models are deficient in not considering all the complexity really that sits behind those three different factors. But even in this paper, there are complexities in this interaction that isn't fully considered. One of which was pointed out by Brader and colleagues, and again at this conference there was quite a lot of discussion about this broader pathobiome and a microbiome associated with plants that also affect how plant diseases develop. And in that paper, Rader and colleagues actually took the step of redrawing that disease triangle to add these other biotic effects. So I think that's quite useful in helping us conceptually think differently in a structured manner about disease development and how we might manage it. And again, I think there's been so much exciting discussion on this topic at this conference. But many others have played around with this concept of the disease triangle or the pyramid. In this case, Castro Moretti and colleagues actually take environment, put all of that under the abiotic factors, and combine the pathogen and these other biotic factors on one of the, they use the points of the triangle at this point, and they add this element of time, and that's something that's been done by others as well. In fact, in Agrios, if you look at the chapter on epidemiology, that element of time is also added in there. But there is this element that Agrios also adds in that epidemiology section that we typically ignore when we talk about the disease triangle, and that's the human component. And Claire, I think much of your talk also referred to this interaction with the farming community, with policy, with a complex financing community environment to actually affect disease control. So I want to reflect a little bit on that element of human impacts on plant disease and how we conceptualize that disease triangle or pyramid. Our impacts on disease development is nothing new. This is a paper out of the 1970s, and in fact you can trace this back to, not trace it back to, but Paul van der Bijl in 1926 reflects also on these human actions that have such a big effect on how diseases develop, how epidemics develop. But in this, Yarwood in this science paper says we've made all of these spectacular advances in terms of production in our agricultural systems, yet these same activities have favored the incidence and development of plant diseases. Again, my co-author and Bruce MacDonald a few years ago also reflected on how our agroecosystems actually stimulates the rapid emergence of plant pathogens and calls on a redesign of these that incorporates or evolutionary biology principles to manage these and not just a simplistic approach within monocultures. Again, at this conference, there was quite a bit of discussion on how this is being implemented. So not new that we're reflecting on our major impact on the development of plant disease. So as I was writing that commentary, I tried to capture that in this disease pyramid and thought it was useful to then add this element of society. But in doing so, I became myself aware of a body of work within sustainable development that is actually in a very structured manner incorporating thinking about the interactions, the really interdependent interactions between ecological processes and social processes in terms of us moving to a more sustainable management of our environmental resources. So here the authors are reflecting on sustainable development goals in general, not plant pathology specifically, but for me as a plant pathologist, looking at that, reflecting on what I've just reflected on with you, that made a lot of sense in terms of also how plant disease management is and should be done, and wondered to what extent these concepts would also be applying to plant pathology. So started a little bit of digging into the literature. So this is from a review on these, the literature on these social ecological systems, these interdependent biological systems. And interestingly, it comes out of the medical profession, microbiologist that first coins this term for the biological, the bacterial pathogens in human systems and social systems that really are intertwined if you think about managing these disease epidemiologies within human systems. In this paper, they track how this concept has been developing since 1998 when it was first coined, and they, in that paper, go up to 2016. I've looked at the Web of Science, and it's around 2,500 articles a year that specifically deals with this concept of intertwined social and ecological systems that affect the functioning of both. Interestingly, more than half of those actually refer to agricultural systems. So agricultural systems are very actively studied by the community that does this, but almost none of those deal with plant health issues. So if you try and link pathogen or pest work into that, very, very few. And I think that is a missed opportunity. There's a very rich body of literature that look at how one could model the various interactions and outcomes emerging from resources, resource uses within this, governance systems, users, and all the factors that influence them, that plant pathology could very readily be using and simply bringing our understanding of the biological processes to this. I'll give you two examples of where this has been used. In this study, they are modeling both the pest dispersal ability, the population growth rates across a landscape, but also the farming community. So how information flows between these different farmers, the heterogeneity within these farming communities. And interestingly, from this model could show that passive diffusion of knowledge, so learning groups that accounts for heterogeneity within that community, is much more effective in actually helping to control the pest than active dissemination of information, so sharing of pamphlets and so forth, and in effect that they could show afterwards as well. There's a very nice potato late blight example. In this model, the green elements are the biophysical, the orange elements are the human factors, and could show that change in market demands has a much bigger effect on sustainable management of potato blight in the system than things like fungicide control on the fungicides. So I'm moving to the end here to say that something that's really appealing within this body of literature is its action-based approach. Structured approaches to define what these social ecological systems might look like, the actors that act within them, and ways in which to parameterize that, measure that, decide on actions, implement actions, and then evaluate those actions. So I think one of the key opportunities for us as a plant pathology community, I think, is looking at these approaches that's very common or increasingly common within sustainable development, and looking to see how we can apply that to our systems. Interestingly, plant health is one of the concepts already dealt with within that. So to finish off, I said in the beginning, and I think plant pathologists understand this intuitively just by the nature of what we do, is that there is an integrated approach necessary to deal with the plant health crisis that we face. And I think it's worth reflecting on how we present this as a community. So if we think about this disease triangle, I think it's useful to reflect the complexity of how we now understand pathogen systems to work. So all of the biotic interactions is not just the pathogen on that line, the abiotic influences and the host, but importantly also that the social ecological intertwined systems has a very big effect on how pathogens emerge and ultimately on the development of epidemics and the management of epidemics. So I think we understand that this integrated approach is necessary. I think the One Health concept that is the theme of the conference, really, this is embedded in it. And I think it's useful to think about how we represent and communicate that. I think as we look to ICPP2028, I think it might be useful. I mean, this concept was infused throughout talks at the conference, just the previous talk, I think the same principle. And I think maybe we can be a little bit more structured in how we engage with those modeling and development approaches. And then as I've said already, I think it's useful to think about how we communicate this in using something like a disease triangle to reflect really the complexity of what actually is impacting our disease development and epidemics. Thank you very much.
Let's see, we have, oh, we'll take Cindy in the room.
Yeah, it's coming.
Oh, thank you.
Thank you for this stimulating talk. So your SOS models, I understand how they're useful post hoc analysis. How can we use them in a predictive way, especially for systems like we don't know where to go, we're just starting out?
Yeah, I should say I'm a biologist myself, so yeah, this is a question that we really should ask the social scientists that really understand and work with these models. But in my reading of the literature, engaging with these stakeholder communities, defining what these social ecological systems look like, actually help you identify elements that might have bigger effects from action than others, and actually might direct action and not just be used to measure it after the fact.
So you kind of open the door for, I'll just give a plug for the roundtable on impact this afternoon, where we're going to talk about some of this prospection that we can do,
exactly.
Tools for looking at the future.
And just and just to say, you can imagine that I was quite inspired by your talk earlier in the conference in the context of what we were doing here. So.
We have a question. Any more in the room? We have a question here that you can also probably follow up with on the app. Have you looked at what is proposed by our colleagues in animal science on ecological immunology concepts?
I can't say that I have looked at that myself, but as I've mentioned, you know, there really is a very rich body of literature that is considering human epidemiology, animal epidemiology, and very actively plant agricultural systems as well, that I think we can very fruitfully draw on.
Okay, that's
Thank
all
you.
time. Thank you.
So we're going to move to the next talk on global challenges facing plant pathology, impact of scientific advances by Michael JEGER. Sorry, Mike. I know him since so long.
Okay, well, thanks very much. Let me mention to start with that this represents the work of a lot of authors who are involved in various publications, which I will go through in a very general way. But nevertheless, this presentation is based in part upon a paper that we published in 2021, the same authors that I've just shown you, which dealt with how you need multidisciplinary approaches to meet some of these global challenges facing plant pathology. Just an aside, we've mentioned the Sustainable Development Goals, and of course much of this paper was aligned towards that particular theme. I might say that there's a meeting at the United Nations in a couple of months' time where really the view is that there's a need to recalibrate some of these Sustainable Development Goals, but that's a matter for discussion at that particular meeting. So this just represents, in a multidimensional way, some of the issues that were approached within this particular paper. Now, I couldn't possibly talk about all of these in this presentation today, so I'm just going to pick out a few of those challenges, and it's interesting that if you look at these particular topics, they've all been represented in one way or another at the sessions and the presentations at this particular conference. But I'm going to start off with looking at cropping systems and genetic diversity. And I'm going to do that at two levels, a whole systems level where you look at biodiversity and ecosystem function, and then I'm going to look at it from the whole plant point of view, where we're comparing resistance to pathogens and what I term as tolerance of disease. So firstly, looking at the whole systems level. Now, please ignore the top panel A of this. I'm afraid my skills with manipulating PowerPoints is not great, unlike many of the presenters here. So just concentrate on the bottom part of this particular slide. And it shows you that if we're moving towards a more sustainable agro-ecosystem, then inevitably, from the state we're in now, there will be certain stresses imposed in trying to make this move. And those stresses can be either physical or they can be biological. So the physical stresses would include nutrients and water because you're trying to reduce their usage. If we're talking about the biological stresses, it's herbicides, pesticides, and other chemical inputs, for example. So how do you mitigate against these stresses that are developing? And the example that's discussed in this paper is the example of P-aphid, which is a vector of viruses in peas, and a pea barley intercrop, where the various mitigation measures that you can put in place to enable this transition are looked at in more detail. So this particular paper can be generalized much more broadly to how you make best of the biodiversity ecosystem function as you're moving to a more sustainable agro-ecosystem. Then at the whole plant level, surprisingly, I find there's been very little discussion of tolerance of plant disease or of abiotic stresses in general at this conference. There's been much on the genetic basis of resistance to pathogens, but very little about how that could be integrated with tolerance of disease. Now, I know many plant pathologists and epidemiologists are suspicious about tolerance because they think it provides a reservoir for pathogens. But we've really got to think about this in a much broader sense. What makes a plant more tolerant to the whole range of biotic and abiotic stressors, and how you can better utilize this. And this just shows how it can be expressed at any stage, right from the germinating seed right through to the next season's production of seed. Tolerance can be expressed. Disease detection and diagnosis features heavily at this particular conference. And there are two elements I wanted to make a few comments on: high throughput sequencing and surveillance. Now, these two elements are rarely looked at together. They remain in their boxes. Now, high throughput sequencing, of course, is come into prominence recently. It's been trailblazed particularly by virologists. They were the first in the plant pathology community to make use of these procedures. And guidelines have been produced. This is a recent publication which takes you through all the way from test set selection to validation to routine implementation. The most important thing is interpretation. And that is a key thing if you're going to make use of these techniques. And often you find that the agronomic or the ecological or the context in which these techniques are being used is simply not made available to those who want to make use of these new techniques. It's been described as you have these new technological advances, but you still have a biological desert in terms of how they're applied. That's controversial, but it's been stated. Also, well, the other thing I wanted to say about that, of course, is the huge amount of data that's generated by these techniques. Are we really in a position where we've got the technologies and the techniques to fully make use of that? There's huge data sets which have been made available. And the same can be made for the advances that are being made in plant disease sensing, whether we're talking about remote sensing, robotics, or other techniques, and the applications that they're put to play in. This is a particular recent paper. The talk on this topic will be given tomorrow. And it looks at, this is a version of a Venn diagram that's a little bit more complicated, but it shows the applications of the dimensions of plant disease sensing. And again, huge amounts of data are being generated, but is it really being put together in such a way that it can be used effectively in terms of disease management? Or spatio-temporal dynamics of disease, this is really what drives disease risk and disease prediction. And I'm going to talk about two examples, one a more ecological one, an invasive pathogen, myrtle rust, and an emergent pathogen, Xylella. So firstly, myrtle rust. Now, myrtle rust appeared in New Zealand in 2017, and the area shaded yellow is the area potential distribution within New Zealand done by knowledge of the pathology of the disease. Now, the dots shown in green are where there are potential ecological refugia, where vulnerable Myrtaceae species could be grown in refugia. Those shown in blue are potential areas which could be restored to more of a native refugia, but are currently arable land of one form or another. So what was done by this particular group in New Zealand was to map out the potential, the determinants of what drives the Myrtaceae species distribution in terms of annual temperature and annual precipitation. And then overlaid on top of that are two scenarios. The one shown in orange is where the maximum infection risk would occur, and in brown, more across the range of the pathogen. And you can see there are potential areas, certainly, that can be defined in terms of where you could site these refugia. And the challenge there, of course, is it's an ecological question. We're talking about regenerative agriculture. We're talking about other means of restoring ecological wild spaces. To what extent can these sorts of techniques be brought to bear to do that? Then moving on to Xylella. There will be a talk given on this, I think, tomorrow in the modeling session. This is, well, Xylella first appeared in almond in Alicante in Spain, again in 2017. And the whole area here is a demarked infected area over that particular scale. A study area was used, which in itself still included 30,000 almond trees. And what was looked at was how you could use a model to see what are the important factors in the spatio-temporal spread of Xylella. And so a simulation model was developed. It was a compartmental model where you move from being susceptible all the way through to being infected. And the model was 100 independent simulations were done, starting with initial distributions of disease, and it was run for 120 months. And a key parameter was a distance parameter, at which you could say beyond that distance, there was no spatial autocorrelation. And this slide, what it does, it just shows you how sensitive this is to where your initial distribution is. So where you've got a little distribution shown here in black, which is very focal and in a particular defined area, compared to where you have certain dots in other particular places, you can see that the amount of disease that develops in that situation was much more than in the other situation, only depending upon the initial distribution and where geographically it was located in relation to the host plant, which was spatially mapped. So the importance of defining this initial distribution and geographical parameters is key. And again, this brings up the issue of surveillance. How can you gear surveillance epidemiologically so it's going to pick out those spots? Again, many papers on microbiome and holobiome studies. I'm going to look at a more classical soil microbiology study to start with, and then move more into what you can actually do in terms of tree declines. So the microbiome association is known to influence the capacity of the host to deal with stresses of various forms. And the question is, does it offer new perspectives for applications of soil microbial applicants? So the key question is here is not how good are these studies for characterizing and cataloging what's there, but can it be used to actually manipulate those communities for a beneficial effect? And that still, I think, remains an open question. And there has been some work done on oak decline, and where they actually looked at the genes involved in nitrogen cycling. And from this pathway, this path diagram that developed from this particular study, it was found that an ammonia oxidizing bacteria combined with pH was strongly associated with asymptomatic trees where others were in decline. So that does open up a possible way in which you can manipulate the microbiome against the decline. Global change also has appeared many times at this conference. Two aspects, climate change in itself and what you might call global trade, which leads to plant disease introductions. Now, there's been many papers and much work done on individual plants, individual crops. What is still lacking is work which looks at the interactions between wild hosts, crop plants, microbes, and the changing climate. And just one example, this phytoplasma here, Phytoplasma solani, affects grapevine and also solanaceous crops. But these are incidental hosts. The real hosts for the pathogen and reproductive hosts for the vector are bindweed and stinging nettle, two wild hosts, effectively considered weeds. So if you're going to understand the nature of climate change impacts on this particular disease, you need to look at the impact, individual impacts on these individual components as well as the overall expression in terms of disease. Quite a challenge. In terms of plant trade, there's been a whole session on risk assessment here, and there is a move now internationally towards more quantitative approaches to risk assessment. This particular representation here is one which arose through developments within EFSA, the European Food Safety Authority. And as well as taking a quantitative approach, you need to think about what the consequences are of introduction. And some of these are listed here. So it's not just a simple case of what's likely to come. You have to look at the impact, and the impact can be multidimensional. And that can be a real challenge, especially as if you're relying on databases, which can be extensive. How do you interrogate them? How can you marshal the information to make these types of predictions? And this brings up this topic of scientometric research. The advances in being able to do this are really many, and some of them are possibly being abused. I'm not sure. And there are many in this room. I think the next two papers, to some extent or another, will refer to this particular topic on how you analyze the data which is in the literature or other sources of information. I'm just going to mention two examples, the first of which is, if you look at the literature on plant pathogens in relation to risk assessments and other matters, you find two forms of information. You find information on new reports of pathogens spreading or emerging in new geographical regions. For example, the CABI databases provide that. Or you can look at the scientific literature and look at where there are quantitative accounts of plant disease outbreaks and epidemics. And I stress quantitative, not just enough to say minor outbreak, major outbreak, where there are real outbreaks. So when this was done for a particular year, 2021, and the results were compared of those two approaches, it's not surprising there was very little overlap in the results, because a new report of a pathogen would normally precede an epidemic or major outbreak of that species. However, this raises an important point that Roel Potting brought up in his presentation as a keynote presentation. The time lag that exists between a pathogen emerging or being reported and a major outbreak occurring. And this can be, in terms of, for example, regulatory measures, can lead to a delay of 6, 7, 8 years, although it does appear to be improving. So this information from these two approaches will identify limitations in identifying. The second example looks at the different sources of information, the scientific articles by taxonomic group, or the published media items, again by taxonomic group. And what you can see here, this is over a 3 or 4 year period, that in the media there is a heavy relative dominance of arthropods, insects and mites, and bacteria, whereas the other taxonomic groups are really underrepresented compared to the scientific literature. So how do you compare these two sources of information? That's what we want to know. I'm not going to say much about artificial intelligence. I could barely follow on from the presentation by Dan Jacobson yesterday on its particular uses. But nevertheless, I would just point out there's a huge number of applications that are being made across the whole scale of plant pathology, from the molecular to the whole planet level. So to what extent can this be used as a cross-cutting approach in plant pathology? And you can do this by looking at finding a multidimensional research network which enables you to do some of these things. This is just one representation. It could be done in other ways, I'm sure. So I'm just going to put up a final concluding slide. I'm not going to speak to it, but if it generates any questions, these are basically the challenges which I mentioned as I've been going through this talk. So thank you.
Are there questions in the room?
Yeah, Cindy? Let's see. She's coming with the mic.
You illustrate the complexity. So in your second to last slide, you talk about the multi-dimensions that we need to get involved to address the challenges. So my question is, after your years of experience, the one after that, the next slide, with the diagram, yeah, that one. Should those disciplines be together in the same research institute or lab, or do we need to, everyone's working on the disciplines and have a common place to meet? Because this is a big discussion about how we construct research institutes. Do we mix the disciplines and have them work together,
Yeah.
or do we put them apart and then let them meet somewhere?
I know, it's a difficult one. It's not just a question of structure, it's the question of the dynamic between the individuals that are going to be put in that structure. And I actually think there's the managers of institutes and universities are quite sympathetic to doing this, but I still think there is resistance from individuals who are involved in disciplines to get involved. And even in an institute which has a multidisciplinary approach, you still have the different silos for which they come across. And what this, of course, doesn't show is the interconnectedness between these different disciplines. So it's a very difficult situation. But really the challenge is not so much for the researchers and the institutes, it's for the research funders and the regulators to make sure their funding work, which does require that sort of approach.
We'll do this one quickly, and then we'll move on to our next.
Thank you, Michael. My question to you is, can you talk about any insight you may have to address the lag between the disease outbreaks and the first report of the plant pathogens?
I mean, in some particular cases, there is a very good account of from when something was first reported to when how it spread and how it caused major outbreaks. And in Europe, the ash dieback, for example, is a good example of that. But for many cases, if you look at new reports, first you need to often manually authenticate them. Secondly, you might find reports, very qualitative accounts, yes, in such and such a place there was an outbreak, but no information on that, no information what the survey did, how it was conducted. So to actually get a fix on that time lag between something appearing and actually causing a serious problem across the board is really very difficult, but it's needed. Well, in my view, it needs at least a 5-year study to actually tease this out, but I haven't got 5 years, so.
Thank you, Michael. All right, we have Anna Berlin. Go ahead.
Thank you. Let's see if I can open.
Here we go.
Oops. So thank you very much for inviting me to talk about our project. And actually this project comes from my frustration in not being able to communicate with the farmers. So I was thinking, so how can I improve my communication with the farmers, that the science that we do actually gets implemented? So this project is actually also linked with some colleagues in environmental communication. So we are simultaneously looking at how do farmers actually want to get information. But I'm not going to talk about that now. I'm going to talk about the information that we think that science can help farmers improve their crop production. So we have conducted a few systematic maps, and you can see my colleagues, some of them, and there are some more. But these ones have helped me in preparing this presentation. And I'm in particular grateful for Elisa sitting here in the audience, and because she has been the backbone of this. So she has keeping track of all of us, making sure that we are doing what we should. Yes, so I'm from Sweden, and this was originally planned for like Swedish farmers. They are well educated, they have good resources, but they still spray a lot. And the crop protection for them, they think it's costly, so it must be effective, sustainable, and profitable. And as you know, we try to limit the negative of pesticides, but at the same time we want to secure the crop production or the yield. So it's a balance. And we need support scientific support to keep this balance in a good, with a very limited amount of pesticide. So the aim was to identify control methods with scientific support that can be implemented in IPM, and also identify gaps where we need more research and to be able to contribute to reduce the use of pesticides. So we have focused on 3 crops: oats, oilseed, rape, and potato. So what are systematic review analyses? So there are 2 versions. There can be systematic maps, which we have done, where you look at the volume of data, and you can do systematic reviews, where you more look into the different type. You look at like one specific tools, and then you can decide you look more on the quality of the available data. But we have more looked at the data at present and not just briefly analyze the quality of each paper. Yes, so the objectives was to get an overview and describe the volume and nature of the scientific literature available, and also provide a basis for policy when they decide for actions, and also where to put resources for funding or for more field work. So we developed protocol, and we were very enthusiastic in 2018. We're not having done this. We got inspired from the medical side, where they use this type of analysis to get this what do you call it? Like standardized treatment procedures. So we thought, we will do all arable crops, and then we did the searches. And ended up with like hundreds of thousands of papers to go through. So we decided to divide it up, and we started with oats, and so that is published, and then we did the oilseed rape, because that's a very important crop. And then when we came to the next, we developed a new protocol, which is a bit limited. So in the second protocol, we did some changes. We only include scientific studies. There is no grey literature. We also made some changes in the screening procedures, and we also broadened it to a global scale. So the primary question was that what is the scientific basis for the different crops, and then you define a population. In this time we had the crop and the Latin name of the crops, and for the first 2 studies, they were basically, we looked only for studies relevant for Sweden in the different climatic zones that would be relevant for Sweden. Intervention, that could be anything. In the first studies, we looked only for field studies, but in the second, with potato, we also looked for studies in pots, because we didn't identify any RNAi or I think there would be. I was missing something, so I thought we need to broaden this. And then you have a comparator, and in our case it was a control, no control, or between interventions. And then the studies had to measure something, like the yield or the improved lower disease or something. similar. So here is the search method. So we started to do searches in scientific databases and Google Scholar. We did screening, and the screenings are done on different eligibility criteria. And this is very big, but the message here is that the resulting quality of the map depends on how you develop and these selection criteria. So they are very important, but I don't want to talk about them now. I want to talk about the analysis and the result. What did we actually find? So bear in mind that we have some different features or criteria for the oat, oilseed, and potato. But you can see the oat and oilseed rape, there are not so many papers that we had to go through. But for the potato, it's quite a few. It's over 1,000 papers that Elisa has gone through and mapped in our database. Also, when you look at the number of studies for potato, it has dramatically increased the last years. It was a lot. I almost regret including 2022, but I think it will add information. What we can see, I will only present general. You can go to the publications and see more detail. But what you can see is that the production and importance of our crop often correlates with the number of studies. So the map shows, the colors in the map shows the production in tons for the different countries, and then this very small figures show the number of studies that we identified are included in the systematic map. Here are which type of disease organisms cause disease. You can see it's in oats and oilseed rape, it's mostly fungi, but in potato you have oilseed that's causing a lot of problem. And for the most common diseases, here I tried to color the different type of pathogens. You can see it's mostly fungi, but you also have, in potato, of course, you have the oilseed, you have some bacteria and virus. If we go for late blight, for example, so here in the top corner you see the overall number of studies, but in the big map shows where the studies about late blight origins. So you can see that it's also differs across the world. For oilseed rape, we divided the different studies based on tenths of years, and you can see that some pathogen and diseases, they were only studied in 1 decade, whereas others have been studied in all. And the most studied disease in oilseed rape is the blackleg, and you see here 2 errors, and the first one is, in the bottom one, which is most common, is Leptosphaera maculans, but then you have Leptosphaera biglobosa, who is also catching up as an important pathogen, causing the same disease. For the control methods, resistance is the top most important control tool. And then you have fungicide and pesticide. I think it's important to note that biological control seems to be up and rising in oilseed rape and potato. And if you look at the number of studies in oilseed rape, we identified 25 articles, and in potato we are 374 and more potatoes studies reporting on biological control. So what does this have for, what can we learn from this? So what we haven't seen really in the data that I presented, but there is a large amount of field-related research that is done by companies, and that's not really available to research and openly, and that is a problem. Because if you want to have something that is implementable directly to the farmer, it also needs to be tested in the field, and not only in a pot. Resistance is important in IPM. I think what we also seen is the ban of pesticides create new problems. We have in sugar beet, for example, has created a new problem in Sweden. That's needs to be studied. We also, in the oilseed rape, there were only 3 articles of the virus, the aphid vectored viruses, and that's and they were from a few decades ago, and that is expected to increase in importance in warmer climate. So there is some knowledge gap that we need to address. So in conclusions, we think that systematic maps can be used to identify both the knowledge and the knowledge gap. And they have a great potential to support plant health. And it can be used like an encyclopedia for selection of different control methods. And we think we identified several interesting research questions that highlights diseases that may become important. And in general, you can see based on the map where the studies come from that the research in plant protection is often based on the importance and availability of funding. And that's linked to the importance of the crop. So it's a very applied science. But I think at the same time, we need the basic knowledge to be able to answer these questions. So it's not a one-side problem. And with that, I would like to say thank you to you for listening and to the funders and my co-authors.
Our regular
Maybe
questioner.
you should keep the micro with you. That's okay.
Thank you for this very nice talk. I understand that you did your literature search and mapping manually.
Yes, we did. I mean, of course, there are some tools that you can use, but in the end,
have you thought about text mining? I mean, because we have a
yes.
big.
I mean, we tested some of it, but of course, some things were done not manually. But
we have a program where we're really investing in that, and to be able to read you wouldn't be scared to include 2022 or all the arable crops anymore.
No. I mean, we tested it, but we thought that we wanted to read and check some things. Oops.
Oh, yes. Here we go.
I can probably shout it. The funding, as you said, is often linked to the importance of the crop, and that has a certain influence on how much money is available. That means many people are working on major crops. No one is looking at minor diseases. And many plant diseases ought to be solved when they're still small, while we still can solve them, instead of following the money. So I think that model is actually seriously flawed. We should work on many more emerging diseases and try to control those while we can, instead of waiting for the funding, because that time lag is way too long.
Yeah, I can only agree, actually.
Thank you very much. So we're gonna, yeah. We're gonna move to the next speaker, who will be talking about data-driven decision, and the talk will be given by Bryony TAYLOR from CABI.
Right, thank you very much, and thank you for having me here today. We were invited to talk in this session on impact very kindly by the organising panel, and I'm here to talk on behalf of my colleague Anna Szyniszewska, who is the technical lead for the Global Burden of Crop Loss programme, who couldn't be here this week. And I'm also talking on behalf of our technical working group and our colleagues who've been working on Global Burden of Crop Loss. My name's Bryony TAYLOR, and I'm the coordinator for modelling and data science at CABI. So, as this session is on impact, I thought I'd give a little bit of background on CABI and who we are as an organisation. We're an international not-for-profit organisation, and our remit is to extend information and apply scientific expertise to help solve problems in agriculture and environment. So by not-for-profit, every profit that we make in CABI means it's reinvested in reaching those goals. So our main aim is to make impact and help change people's lives. To give you a bit of an unusual spin, we're a member country organisation, so we have 49 member countries, and these are the flags up here, but that does not restrict the countries which we work in. But what it does do is put us in a unique situation where we talk to our member countries every 3 years at our review conferences. We learn about their problems, they tell us what they need, and we write our strategy as an organisation to help meet those needs. And it's a very important concept for us. And they pay a nominal fee to be part of CABI, but we take their needs and we turn that into donor funding and large projects and programs. As an organisation, we have a large global reach. We have centres all around the world on the yellow dots here, but we also have people placed in different countries in the world. So as an organisation, we have strong links into each of the regions within which we work, and we work really closely with the governments, the agricultural research organisations, and we have a real way of driving impact through right down to the people who need the information, be it at a governmental level or at a farmer level. So we were invited here to talk today about our Global Burden of Crop Loss programme. And this is a programme that's meeting a need that we're all aware of, that we're still continually losing crops for various reasons around the world. And this is becoming an increasing challenge under the umbrella of climate change. So we're seeing sporadic weather events which are causing losses. We're seeing changes in the phenology of our crops and our pests and our diseases. And we need a data-driven way that is able to track how things are changing and attributing losses. So from the donor's perspective, the calculation that we've made is that there's $25.8 billion that is spent annually on agricultural research and development. But if we don't know what is being lost, where and why, how do we know that those investments are being made in the correct way? So our remit is to provide information that is easily interrogable by these people and other people who need the information to help direct investment into the right places. So the global burden of crop loss is strongly aligned with the global burden of disease and the global burden of animal diseases. And the way that we are set up is replicated on the format that was used by the global burden of disease. So in this programme, they brought together hundreds of billions of data points, and put them together into an easy, interrogable way of interpreting the information around what are the causes of human morbidity. And the global burden of animal diseases are doing this in a similar way, and this is being led by the University of Liverpool in the UK. So, what are we trying to achieve with the global burden of crop loss? We are trying to provide an authoritative and rigorous evidence of impacts, causes, and risk factors on crop loss. We want to produce evidence-based estimates of crop loss and attribution to direct funding, policy, and help highlight where research is needed. As you can imagine, this is a great challenge, and our mission is to do this, our vision is to do this in a data-driven way, and our mission is to make sure that we do this in a collaborative way and involve many partners and the best of cutting-edge scientific research. So this is the framework that we're working towards, our conceptual framework. On the left-hand side, it is the left on the screen, that's good. You have attainable yield. So this is attainable yield in context. So what is possible for someone to grow in a particular area given their weather conditions, their water availability, their nutrients, and their socio-economic factors. And this is generally a modelled factor. When you compare this against what is actually produced in an area, you come up with a loss envelope, and this is a value of losses or an amount of losses where we know the proportion of losses that are occurring. But we want to help split this down into biotic and abiotic factors. So under the biotic are obviously the pest and disease prevalence, but under the abiotic factors are shock events like wildfire, flooding, that type of thing. And we want to put a value against those losses. But of course, to work out the burden estimate, we need to look at the cost of the control measures. What are the inputs that are being put in? And how do we sort of balance the books and show, well, actually, what is the total burden? And these are the ways that we're trying to achieve this. So, as this is an impact session, I'm not going to dwell too much on the sort of technical side of things, but this is kind of our overall theory of change. So at the top, you'll see that we want to reduce crop losses, and the impacts will be improving food security and resilience for farmers globally. That's where we want to be. But to get there, we have to start at the bottom, and we have to look at the inputs of how we do this, which are the data sets, the methodologies, the skills, expertise, and the collaboration required to do this. This will produce the outputs, which will be these loss estimates, and then these will be shared and communicated to stakeholders so they know where and why losses are occurring, with the long-term outcome that we will have appropriate resource allocation, and national and regional strategies will be able to embed this information, and the funding is going to where it's needed most. And of course, this is all embedded in the Sustainable Development Goals, and the ones that we are looking to support and provide information for are here. And I won't dwell on these too much, just in the interest of time. But in the questions that were put up at the start, we were asked to talk about the challenges. And I think this week, through many of the sessions that we've been to, we know that there are a lot of challenges if you want to have a data-driven approach. So the first challenge really is around the collaboration. How do we all work together? And how do we govern the collaborations? If we're having scientific research feeding into an overall umbrella of the global burden of crop loss, how do we govern that and make sure everybody's represented? And how do we make sure that this program is sustainable and not relying on donor funding? Data, obviously a massive issue. There's lots of data in disparate places. There are many places where data is unavailable. So how do we make that happen, and how do we make that change? And I'll address some of this in our next slides. And communication, how do we make sure that our results are communicated effectively to the people who need to get the information? And of course, at a country level, where we're receiving or putting out these estimates, how do we ensure there's data governance that keep the national interests at heart? We're not producing any information that's going to harm their economies in any way. So these are the challenges that we're considering and the ones that we'd like to overcome. So progress to date, we've set up a technical working group, and this is a collaboration across different universities and research centres and organisations where we are exchanging ideas about how we overcome some of these technical challenges around data and collaboration, and how we answer some of the pressing questions within the global burden of crop loss. And obviously data, I think Cindy, you just mentioned this. If we want to produce replicable methodologies that produce ongoing estimates of crop loss, we need to look at how we're sourcing our data. So we've been working on text mining and natural language processing with Rothamsted Research to look at seeing how we can build pipelines to extract impact data automatically from the literature. And the next step will be to look at large language models as well to do this. But it's important that we're able to access and aggregate information in an efficient and replicatable manner. So in terms of communicating our results, to date we have been working on proof of concepts through funding from various sources, which will be acknowledged at the end. And we've got attainable yields in context. And these are global gridded crop maps for maize, wheat, soya, and rice. And these are based on the parameters that I talked about earlier. Lost my thread of thought. And then these are compared to the yield loss envelope. And there are technical challenges around estimating the attainable yield in context. What you find is a lot of the models are correlated to production figures, which then affect like your yield loss envelope. So we're looking at aggregating data from field trials, and we have done this, aggregating data from field trials around the world to then use actual scientific research data to validate and build the models so they have a more representative representation of the attainable yield. And also, when you're looking at the abiotic losses, you have to look at pest prioritization methodologies. When you have a list of pests, so I think a lot of the talks that I've seen this week have been using the CABI data where we have big lists of pests for the countries, but if you download, say, one country, it might have 200, 400, 500, 1,000 pests, and how do you work out which of those are most important? So the work that we're doing at the moment is working across programs at CABI, and we've had other programs that have done expert elicitation of what the key pests are in some of the countries in sub-Saharan Africa, and we have lists that we can use as a validation to work out a more automated way to do pest prioritization. And at the moment, these exist in our shiny dashboards, and these are more of a demonstrator tool to take to donors to talk to them about the information we're producing and see what kind of information they actually need and how they need to have that information presented. We're also working in the wider context of One Food. So we have a program that's funded by CFAS, which is a UK organisation with the same name as CFAS in Europe. But this is looking at actually how do crop losses link into the wider food chain. So yes, we've got food that's going to that's being lost, but then what happens on the rest of the food chain? How does it go through to animal feed? How does it go through to markets? And I think there was someone said earlier about how markets are very important affecting production. So at the moment, this is a conceptual framework, and we have a 2-year project to put real figures into that. And we're also looking at the wider context of One Health. So how does this information embed itself in the One Health framework? And again, this is under the CFAS funding. And we have a paper that we have to submit next month to the World Organization of Animal Health, showing how we're working really closely with the global burden of animal diseases, and we're aligning our methodologies and the way that we work together to really get this One Health perspective. So we're trying to get impact by working in a broader playing field, as it were. But I want to come back to pathways to impact for the last part of the presentation. We've got these member countries, and we have ways to communicate our results back to them and really sort of gauge what the issues are. And through other programs at CABI, we have the PlantWise program, or PlantWise Plus as it's now called, which has had incredible impact all around the world. We've trained over 13,000 plant doctors who have been trained in identification of pests and diseases and give advice to farmers. And the farmers that we've reached are in the number of over 54 million. So we have a real route down to the ground level of how to disseminate information. And through other programs, we have disseminated information through the Plant Doctor Network to actually give advice to farmers and help improve their yields. And this is through baseline and end-line monitoring and evaluation efforts. And there's a lot of information on our website about the impacts that the PlantWise Plus program has made. So we're really fortunate to be embedded in an organisation that has so many routes to impact. So our objectives for the next 5 years: we'll be working on that under the bonnet. How do we estimate those wheat losses and economic burden globally? And this will be extended across 5 crops, I think 3 of which I've mentioned. We want to continue working to expand the technical working group, and we want to continue to link in with existing initiatives. It's really important that the data and the information flows that we produce can be used by other people and create onward impact, not just through the policy and donors, but into other scientific programs. And we want to increase those high-level collaborations between partner organizations. We want to make sure that we're looking at governance, and we keep governance well covered as we go through. So that's both in terms of collaboration and in terms of the data that we use. And in terms of data and modelling, we want to create a database that has reported impacts on crop pests from both the published literature and field trial data to really underpin the attribution modelling. And in terms of communication, we want to publish our estimates in peer-reviewed journals, which we are working on, and we will develop an online tool which will allow users to explore the estimates in the similar way to the global burden of disease. And this is the road that we're on, and I think that this program is something that has really thought about impact from the start and like how we extend that, and we're really proud to be part of it. So I just want to gratefully acknowledge all of our funders, both our core funders at CABI and people who've supported global burden of crop loss, and of course our technical working group. Thank you.
have any questions in the room. We have a couple on the app, but we can hit the room first. All right, so thank you. Someone wanted to know, do we have common ontologies in plant pathology to make a common database?
So we have the CAB thesaurus, which has common ontologies, which hopefully is one of the best thesauruses in the world. I don't know the plural correctly, but really they do mine down right down to the important ontologies in every aspect of agriculture. And throughout all of our programs, we're working to make sure that the same ontologies are used so we can have interoperable data across all our programs. So GBCL will be one of those.
Another similar question, are there websites like Geo or PubMed, but for field data results?
Sorry?
Are there websites like Geo or PubMed for field data results?
Yes, so the CG centres, they've done a fantastic job with their data, and they have the Guardian portal, and they've published a lot of their results in an open access format. And there's also other databases, which I will have to ask our technical team what they are. But there's a lot of information that's coming from Earth observation information as well, and there's different layers of information that are being used. So.
Any other questions in the room? There's a good comment someone made, a 3-point comment, which I think everybody can just log on to this, the app and look at, because it's not specifically about yours, it's about the whole concept. Anything else? All right, thank you so much, that was great.
Thank you very much.
And next we have Toussaint Hussein, who unfortunately couldn't get his visa, so we will be playing his video that he recorded for us. Maybe you should go. Oh, well, maybe I have to click it. And he's unable to answer questions, obviously, so we can take a couple questions more generally after. Maybe I have to click.
A very good afternoon to all of you, respected chairs, on the dais, off the dais, and my dear friends. Myself, Dr. Tausif Hussain. I'm from India. Today, I'm going to deliver my talks on the novel plant growth promoting rhizobacterias as a potential indicator of sustainable agriculture development. Human health is closely connected to to soil health, because soil health is crucial for producing nutritious food, which is essential for our well-being. To ensure human well-being, it is important to prioritize sustainable soil management practices that promote soil health, such as the organic farming, crop rotation, cover cropping, and minimizing the use of harmful chemicals. It is a key to tackle the climate change as it captures and stores the carbon. It is a life support system, and soil is the foundation of an elegant symbiotic ecosystem. Regenerative agriculture is one of the approach to the sustainable soil management that is rapidly gaining attraction nowadays. So every day around 2,000 farmers in India are considering to quit the farming in the recent years. They are in piles of debt due to unmonitored chemical uses in their soil, as soil is losing fertility, and extreme weather conditions are massively affecting their annual crop. Because in Indian farming system, the excessive chemicals are widely used to enhance the growth of the plant, because chemical fertilizers act as a fast food which causes the plant to grow speedily. However, at the same time, creating a negative impact on human beings. For example, in the state of Punjab, around 246 kilogram of fertilizers per hectares are used in comparison to the national average of 135 kg per hectare. This indiscriminate use of these agrochemical fertilizers has taken a toll on our environment, resulting in irreparable ecological damage, soil infertility, and a toxic food chains in India. Our laboratory was established in 2019. One of the main focus is the soil testing because it helps in optimization of crop production, diagnosis the nutrient deficiencies in the soil, which is an important practice to manage the fertilizers application. So I joined in 2021 here as a plant pathologist. So looking at this gap, I launched this initiative of biological based formulations having the natural bioproperties, that is the phosphorus solubilizing, potassium solubilizing, and biological fixation activity, because these formulations can help to maintain balance the pH of the soil, that is widely it helps for the optimum growth of the plant. So during this study, I collected some soil samples, specifically from the rhizosphere areas. These samples were collected from the extreme environmental conditions. For example, there is no human interventions. The temperature is very high and there is a high humidity, and it is full of rich biodiversity. After that, I go for the screening isolation of these isolation of different media, having for example, like Alexandro agar media for the potassium solubilizing, Pewaski agar media for the phosphorus solubilization, and for the biological nitrogen fixation, we have used the ammonia nitrogen free media as well as the ammonification test. After this isolation, I found that there are a couple of bacteria which has been playing a very important role in the phosphorus solubilization, potassium solubilization, and the biological nitrogen fixation. You can see here there are three bacteria, that is W, Fa, and Q, have demonstrated a very high potassium as well as the phosphorus solubilizing index here. So later on, I have also characterized through the biochemical as the molecular characterization. We found that this bacteria found to be the most interesting one. It is the Bacillus japonicus is there, then Diella giensis and Agrobacterium tumefaciens, because this Diella giensis is one of the most first time being reported from India, and Agrobacterium tumefaciens, this is also a very this bacteria has also demonstrated very promising results specifically in case of nitrogen fixation, and there is no reports till date regarding the tumor formation from this bacteria. So using all three bacteria, we developed very cheap and affordable, using the organic amendments in this, develop a bioformulation. We carried out some seed germination bioassay test here. You can on different crops like mung bean, Bigenia radiata, and Cicer arientum, or the wheat. You can see seeds that these seeds were soaked for 30 minutes in this formulation on a moist filter paper. The vigor index was 3 times more higher, and highly encouraging results were noted in comparison with the uninoculated one. Similarly, a biofilm property feature was also noted down from these bacteria. You can see from this slide. We use a standard traditional method for using crystal violet dye, and after the overnight keeping it overnight, we found that a ring formation was noted down. That is a key feature of the biofilm formation. This biofilm property has added a unique feature to our product. This is the last year of results on the cotton crop. You can see the effect of the sebum biofertilizers on the cotton crop. The more increased number of birds were found here and higher production and crop productivity. In the cotton crop, the formulation was sprayed through the foliar spraying, that is 5 ml per liter. Again, we also carried out the soil health analysis on after 15 days of interval. You can see we have carried out this soil analysis using the ICPMS from manufacturer from Thermo Fisher. A very quite high increment in nitrogen and phosphorus amount was noted down using our own sebum biofertilizers in comparison to the other products that is the top selling product in the Indian market. However, potassium amount was nearly increased similarly in the 30 days. And this is the last just 1 month old and zucchini summer squash. You can see the results of the foliar spray on the row. There is a increase in the growth as well as in the root biomass here. And this is our product that we have formulated and developed here, having the pH of 5.8 and CO2 of 8.1, and it can sustain the temperature from 10 degrees to 50 degrees Celsius. And this has been made using the organic amendment here. And each element and functions as a part of a delicately balanced integrated system with the plant genetics and the environment. So one of the most important step is the commercialization of the product is the next step we are looking forward. And we need to achieve this impact, however, need to do a large scale field trials at different agroclimatic locations. Last but not the least, we need the funding and the structure and the team efforts here, a good working culture, working life balance that we are focusing here in the present condition going on here. Thank you so much. Thank you for the ICPP for giving me this opportunity to present my research finding, and for all the jury members, thank you so much.
If we can't do Q&A with him, obviously, but if anyone wants to make a comment or about anything from any of the talks today, feel free to. Otherwise, we can just be dismissed for lunch. Oh, yeah. Yeah. I mean, you can't isolate any of the components. Yeah. I mean, you mentioned that they're going to be reevaluating the SDGs at the Global Assembly next month, and we use them because we're part of various UN programs, but there's certainly issues with them because they take the whole. They de-silo everything, but then I also don't know anybody that works in impact that uses one SDG. They all use handfuls of them. So I'll be curious to see how they adjust them. I feel like mostly those SDGs are markers of what impact you're already doing. It's not goal-driven, even though they were designed to be goal-driven. So like we were selected to be represented at the General Assembly for Life on Land, which is SDG 15, and there is one indicator within that that's on invasive species, which is not like, that's like the 4th or 5th SDG down the line that we would ever pick, but they invited us because I don't know why, but it didn't change, like that particular SDG doesn't change what we're doing. It's all the other things that are changing and motivating what we're doing. So there's definitely some flaws in the system of impact measurement because they get separated out so much.
So we wish you a good lunch and big thanks to all the speakers.